In this final video for chapter 14, we're going to kind of wrap up some odds and ends. Um, and so the first thing I want to talk about is, if this is the adult brain, right, in all its complexity and amazingness, like how do we get to this point? And so let's talk a little bit about the development of the brain, the embryonic development. And so when you hear people talk about brain development, right, they will point out that we start out as this, what they call the neural tube. And we can go back a little further, right? But notice here these primary brain vesicles, what they're calling them, these little maybe enlarged areas of this tube, they're starting the development as early as 3 weeks after conception. Which is pretty remarkable. And really what we have is we have 3 different regions. The most anterior is called the prosencephalon, or forebrain, the mesencephalon, or midbrain, and then the rhombencephalon, or hindbrain. A little bit later, here we are at 6 weeks, we start to see that we get even more divisions, right, in these enlargements. And so what we're going to see happen is the prosencephalon splits in 2. It becomes the telencephalon, which is destined to become the cerebrum, as well as the diencephalon, which we talked about previously, and it continues to be known as the diencephalon. All right, so that one's a little easier. The mesencephalon does not divide into any secondary brain vesicles, right? It stays the mesencephalon and then becomes the midbrain. The rhombencephalon splits in 2. We get the metencephalon, which becomes the cerebellum and the pons, as well as the myelencephalon, which becomes the medulla oblongata. So all of these major brain regions that we talked about in the adult brain come from a variety of enlargements in these tubes. If you take it back even a little further, if you recall from the chapter on tissues, we said that the nervous system actually comes from the ectoderm. Right, we had 3 germ layers in the embryo. The outermost one is the ectoderm. And so this is always a little counterintuitive to folks that this outermost layer would somehow become the nervous system, which seems like so deep and protected. But basically this is what it looks like, right? So if you picture the whole embryo, right, one side has what's called the neural plate. It starts to indent into the neural groove and eventually it folds over, right? And then other tissues are able to come around. And so you can picture this kind of being the vertebrae and the spinal cord going right down the middle. There's an actual picture, right, of that neural plate, how it kind of rolls into this groove, eventually a tube. And here we go, even after the first month, right, it's completely enclosed. And so that's some really important development that happens very early on during embryonic development, which is why alcohol consumption during pregnancy can be quite problematic, right? The neural development is happening early, perhaps before a woman even knows she's pregnant. Okay, and again, just another look at how those they call them dilations, change, right? Prosencephalon, mesencephalon, rhombencephalon, and then they're going to split again. So maybe those pictures, these are from a different text, if they help you. And then we're color coordinated so we can see kind of how that ends up in the adult brain. All right. Couple other topics. So the ventricles, right? The ventricles in the brain are these spaces, much like the ventricle in the heart is a chamber, and you end up with 4 ventricles. Now, the one that tends to throw people is we talk about the lateral ventricles. They don't get numbered 1 or 2, and nobody seems to care really if they're left or right? But you have these 2 lateral ventricles, and then they both connect into the 3rd ventricle. You have a cerebral aqueduct that carries you down to the 4th ventricle. In these ventricles, I think this is a super cool picture. You can kind of picture like 3-dimensionally how much open space there actually is deep inside the brain. Anyway, these ventricles are lined with ependymal cells that are making and circulating the cerebral spinal fluid. And remember, we talked about CSF with the spinal cord as a bit of a protection and also nourishing. Here in the brain, it actually becomes really important. So this cerebral spinal fluid is in these ventricles. It can travel down the central canal of the spinal cord, and then it's also in that subarachnoid space, so kind of surrounding the brain. It actually floats the brain, right? The brain is heavy enough that if it sat on the base of your cranium, all these nerves on the underside here would get crushed. So your brain is actually kind of floating in cerebral spinal fluid. It's also a super important cushion, right? So they talk about it like, right, you have the skull, this outer protection, and then you had the meninges, right? Those are continuous with the meninges on the spinal cord, and they're the same. And then you have this cerebral spinal fluid. And so I think the example they give you in the book is like, man, if your car crashes into a phone pole, like the car gets crushed, right? That's a level of protection. Maybe that's like that hard outer protection, like the skull. But then if you don't have a seat belt or an airbag, you are still going to get really hurt when you yourself run into the car, the same way the brain would get hurt if it slammed into the skull. And that does happen sometimes. The meninges are kind of your seatbelt, right? They're trying to keep things anchored, and the cerebral spinal fluid is that airbag, a little extra cushion, but it doesn't always obviously prevent brain injury. Right, so it's cushioning the brain, supporting, kind of floating the brain, and then just like in the spinal cord, transporting nutrients, wastes, chemical messengers, all those sorts of things, right? It does circulate in that subarachnoid space, right, between the meninges, and we'll look a little bit more. Remember that cerebral spinal fluid, it's being made by the ependymal cells in the ventricles, but then it needs to go away, right? We have a limited amount of space. And so we will see that there's some we call dural sinuses, some spaces that collect that CSF in the dura mater and return it to the jugular veins. Speaking of blood supply, so the blood supply to the brain is redundant. You actually have 4 different arteries, 2 internal carotid arteries, right? So they're showing these internal carotid arteries coming up to the brain. See this internal carotid and this internal carotid, right? One on the left, one on the right. And then also you have 2 vertebral arteries. If you recall, on the spinal column, on the cervical vertebrae, you had an extra hole on the transverse processes. That is for this vertebral artery. So those 2 come together, and so you actually end up with what's called the circle of Willis. And the circle of Willis lets all 4 of those arteries kind of feed together and then act as this redundant blood supply for the brain. And that tells you, as you already knew, the brain is important. The brain is greedy, right? It wants its oxygen. It definitely wants its glucose. And so we've got a rich blood supply to support that. Do recall, right, that all of these blood vessels, when you get down to the capillary level, you are going to have a blood-brain barrier, right? Specialized astrocytes helping to cover those capillaries, and those capillaries are also held together with extra tight junctions. If you remember the tight junctions from the cell chapter, so chapter 3, making sure that everything that leaves the blood and goes into the brain has to actually go through a cell membrane and not through any kind of spaces. You do also have what they call the blood CSF barrier when you look at making cerebral spinal fluid. Okay, let's see here. Let's go to that. So there are some nice images here showing you in the ventricles how cerebral spinal fluid is actually made. And so here's a capillary. And this is trying to show that you also have this blood cerebral spinal fluid barrier, right? So you have a blood-brain barrier and then also a blood CSF barrier. And in this case, it's the ependymal cells that are lining that capillary. They're pointing out the tight junctions. Those are in the capillaries as well as between these ependymal cells. But we're making sure things can't move freely out into the brain. And so here is the example they're showing of the blood-brain barrier, right? The yellow astrocytes and their little foot processes covering the capillary, making sure that wastes can get into the bloodstream and nutrients and oxygen out of the bloodstream, but nothing else. And again, those ependymal cells making the cerebral spinal fluid. To make enough cerebral spinal fluid, notice, we'll zoom in here, but what they're pointing out is that in the ventricles, you actually have these extra, here we go, these extra like knots of capillaries called the choroid plexus. Choroid plexus. And so it's extra vasculature, again, covered in ependymal cells to crank out some more cerebral spinal fluid. All right, so there they're trying to show you that choroid plexus here in the third ventricle. You can see some in the fourth. You'll see this again when you do your dissections. So again, notice these connections here. We've got the lateral ventricles on either side of this membrane, this little wall. The third ventricle kind of surrounding the thalamus. The cerebral aqueduct takes you down to that fourth ventricle, and then we're able to put that CSF either out into the subarachnoid space or down that central canal. Right, we see cerebral spinal fluid circulating. And again, this is where I was talking about that ability to kind of recapture cerebral spinal fluid, right? Here we are, the pia mater directly covering the brain, the neurons, and see how the subarachnoid space is kind of all these little struts, like it holds a little opening. That's full of cerebral spinal fluid, right? That cerebral spinal fluid, we can't just keep making more, we would build up too much pressure in the skull. We can't keep making more unless we get rid of some. And so that's what happens here. We have these little, they call it like an arachnoid villus, a nubbin, where we're actually going to go ahead and dump cerebral spinal fluid into this space between the two layers of the dura mater. So the one big difference with the dura mater in the brain versus the vertebral cavity, the dura mater has a periosteal layer actually attached to the cranium, right? There's no epidural space when you're in the skull. Instead, we have a periosteal layer tightly glued to the skull. And then we have another layer, the meningeal layer, and the space is now actually between those two layers. That space is called a sinus, and we're able to actually collect cerebral spinal fluid here and then return it to the jugular veins. Okay, I think last but not least, let's touch base real quick on the cranial nerves. This will definitely be a topic in lab where you're actually identifying, pointing out the cranial nerves. But for me, what I'd like you to know is the number of the cranial nerve, its name, its function, and whether it is a sensory nerve, a motor nerve, or both. I will lay that out for you. So notice here on the diencephalon and brainstem, cranial nerve number 1 is actually the olfactory nerve, and that does not come in here. So they're starting at number 2, right? And what they're showing are these nerves are cut off, right? Cranial nerve number 2 is the optic nerve, and so one of these nubbins goes to each eye. So it always looks a little odd. You're like, what are these little chunks of flesh? Those are in fact nerves, right? Bundles of axons that have left the central nervous system and are heading out of the skull. All those holes that you learned in the skull in lab, now you can see they have a function, right? So these all have to exit somewhere, and a lot of them have named openings. So let's try this. I don't know if it'll actually, maybe it won't let me do this. I don't think it's going to let me do this. Maybe we'll just have to do it in class. So what I was kind of picturing here was being able to type on a blank page to tell you the names of all the nerves, right, what they do, and then if they are sensory, motor, or both. This is going to be the most boring thing ever, but let's try this. Okay, so no, not gonna work at all. Let's see. Wow, so many slides. Here we're back to cranial nerves. This is what they're gonna look like more in real life. Yeah, I guess we'll make a table in class so that you understand kind of the function of each of these cranial nerves. But know that's coming your way. Maybe you even want to build that ahead of time. Again, the number of the nerve, the name of the nerve, what its function is, and then if it carries sensory information, motor information, or both.